Welcome to the Regenerative Medicine and 3D Printing 5-on-5 session. I'm Bob Goldberg. I'll be co-moderating this session with Scott Hollister, and we have two panelists with us today, Raj Krishnamurthy and Timothy Nelson. Unfortunately, our third panelist was sick today, and that's the bad news. The good news is that's going to allow us to catch up a little bit on time. So without further ado, we're going to go ahead and get started. I'd first like to just thank, if I could have my slides, I'd like to thank Anthony for the opportunity to highlight this particular topic area, which I think you'll see has tremendous potential to impact pediatric healthcare and already is in fact doing so. So there we go. So when we think about regenerative medicine, I think this is the paradigm that a lot of us think about, in which you might take cells out of a patient's body and perhaps expand them and then combine them with some sort of biomaterial that may include some signaling factors in there. And then that whole combination needs to go into some sort of bioreactor, which, out of which would come something that looks like the organ or tissue that you're trying to replace, and then plug it back into the human. And this is sort of, if you will, this is sort of the home run, I guess, that the field sort of thought about from the beginning. And in reality, there have been very few success stories that actually work like this for a number of reasons. There's some technical challenges, for example, the fact that it sort of ignores the fact that all these complex three-dimensional organs and tissues are typically vascularized, and we don't know how to do that yet. We don't know how to make them vascularized in vitro. And there's also some non-technical challenges, like it's really complicated and expensive to do all this. And so how do you build a business model with the reimbursement challenges we have and so forth? So in fact, if you look at what's actually happening, to use sort of a baking analogy, instead of creating the entire cake and plugging it in, we instead have been taking some of the ingredients, like the cells themselves or the biomaterials, and putting them in and learning how to cooperate with the body's own regenerative capacity. And in fact, that's one of, I think, the real advantages that pediatric patients have, is that they have a higher innate regenerative capacity than adults do. So we'll talk today about some of the different ways of doing this, but I just want to give a little bit of overview on the different factors that we're talking about. And of course, biomaterials play a huge part in this. And over the last 25 years or so in this field, there's been just a tremendous increase in the number of biomaterials that we have to choose from in our toolbox. And I roughly segmented these into synthetic type of biomaterials. And Raj and Scott are going to talk about 3D printing, which is one way for manufacturing these synthetic materials. But then there's an entire other class of tissue-based, natural tissue-based derived materials. And these have some advantages and disadvantages to both sides and some challenges associated with them. One of the companies, or one of the examples of tissue-based products that I wanted to talk about was derived from human amnion. So these are donated placental, it's basically human waste from birth, that there are several companies around the US and elsewhere that are commercializing these tissues because they're full of pro-healing factors. In fact, there are over 200 proteins that, if you process them properly, can be delivered into a patient and can help with healing. So this is just a company I work with in Atlanta called Mimetics, and you can get this product in either a membrane form or a micronized form, and it's being used for a lot of different applications, including diabetic ulcers. You can see here an example of a pediatric patient that had a scald injury that received amnion treatment. And one of the interesting things about these biologic treatments that are used for wound healing is they tend to manage the pain better than normal wound healing dressings. So you can see the 1-week follow-up here, the burn is healing well, and at 3 to 4 weeks, the burns have healed quite well, and in fact, the pigment is returning. So one of the reasons that these types of biomaterials are advantageous is the FDA regulation of these materials is actually a little bit less stringent than if you were trying to combine a cell with a synthetic biomaterial. But one of the challenges these companies are facing now is that the rules keep changing. And so you can see here just a new guidance document from the FDA from October 2015 on how the regulation of cells, tissues, and cellular and tissue-based products are being regulated. So what about cell therapies? And I think Tim's going to talk some about cell therapies. There's a huge number of pediatric conditions that may benefit, and clinical trials that are going on in all these different areas. And I think it's interesting to look at the growth in the market, even though there's still a lot of huge challenges to be met for cell therapies, including, for example, if any one of these ends up being a home run, we will use up the world's supply of serum, which is used for expanding cells. So we clearly need to come up with a serum replacement. But you can see that there's been really solid growth in this field, and it's really ready to explode if we address some of the challenges that we have. You can see the clinical trials worldwide, almost 9,000 as of October, and more than half of those in the US. And one of the things that you think about in this field is what is the source of the stem cells. So you can have stem cells that come from the patient, autologous stem cells, or potentially from an allogeneic source from another patient. And those have different advantages and disadvantages. They also have different business models, which are interesting. And you can see from the growth in the clinical trials, both of these are growing. It's sort of unclear which is going to be the most prevalent, and it may depend on the application. And then something I think Tim is going to talk about is about 10 years ago, there was a really exciting discovery in the field that we could take the patient's own cells, adult cells, and use reprogramming, genetic reprogramming, to turn them into something that very closely approximates the potential of an embryonic stem cell. And that has huge potential for both therapeutic applications, but also, as you can see at the bottom, for using those cells as disease models for looking at what the etiology of a disease is, or for drug screening. All of these things are very, very promising, but one of the clear challenges that we have is that we can't make these very inexpensively. And so we've been excited to be involved in a recent new consortia on cell manufacturing, with over 30 companies involved and all the different government agencies, with the idea being to come up with new technologies. We need to innovate really in manufacturing technologies for scale up and scale out to produce a large number of high quality cells. And this is so that this is available to everybody. It's not just available to Kobe Bryant to go off into a foreign country and pay $20,000 for his cell therapy. We want this for everybody. And there are, in fact, cell therapy products right now that are FDA approved, clinically effective, that are sitting on a shelf because the companies can't make money on those. So this is something that's a real challenge for the field and that will need to be addressed. Okay, so just lastly, I just wanted to mention why I think regenerative medicine makes sense for pediatric patients, because I think we generally think of regenerative medicine as something that we might use when parts have worn out. But in fact, there are some great reasons for using this for kids. Children have the highest capacity of regeneration. We need customized solutions, and Scott will talk about that. And we need long-term restoration of function. Scott?
Thanks, Bob. So I'm going to give a little introduction to 3D printing, and one backwards. Get it on the right. So first I wanted, for some of you who may not be aware of what 3D printing is, you've probably heard a lot of buzz in the press about it. 3D printing is really a set of technologies by which we can build structures on a layer by layer basis. And you can think about it broadly in two terms. There are sort of nozzle-based systems where you squeeze material out like a tube of toothpaste onto a platform and move the nozzle and build a structure. And then on your left, you can see there are laser-based systems in which you basically take a laser beam and you process material, you melt it together, or you photopolymerize material, and then you lower the build platform and you build up three-dimensional structures that way. And I'm not sure if the video on the bottom is going to work, but that's an example of a machine in our laboratory that we use to build resorbable, implantable materials using the laser-based approach, the laser sintering approach. So that's how people use three-dimensional printing to manufacture structures. But I think the other exciting thing is that, of course, you have to have something to go into the machine, right? You have to have a design to put into the machine to print. And I think one of the revolutions in the use of 3D printing in medicine is the ability, as shown on your right, to take direct patient image data and to design based on that patient image data. So now we can make patient-specific implants. And in fact, this is an example of a implantable airway splint that we've developed, and we'll show the case later on in a few slides. But to show you the power of combining these approaches, we had a concept with the surgeons on a Wednesday to modify one splint design. We did the design on a Thursday, and then we built it on a Friday, and it was implanted the next week in the child. This just shows you the range of structures that you can build. We've built these all on our laser sintering system from the resorbable material. We've implanted all of these in animal models or human, except for the brain. We haven't done that yet. But it just shows you the range of possibilities. And this is all built on one machine with one manufacturing process. And just to give you an idea of how 3D printing is currently being used in medicine, we've had a number of talks already at the meeting talking about the use of models for pre-op planning, for surgical planning. And obviously, one of the sessions yesterday with the patients and their families, one of the big issues was communication. How do you talk about the procedures? And that's another role that 3D printing can fill, because you can build models and you can use these models to help explain to patients what their procedure will be, how to get patient interaction with their procedure. So it's being used quite a bit for pre-op planning, for surgical planning, intra-op guides, patient education, physician education, and surgical simulation. What we see as the future is actually, and this is coming now, is the use of 3D printing to build medical devices, both prosthetics, implantable devices, resorbable or permanent devices. And as Bob mentioned, this is really a fusion of two fields in which you have 3D printing and regenerative medicine coming together, either printing cells directly or printing the scaffolds on which you will place the cells. So just to sort of wrap up before the movie, I think there's a tremendous amount of promise, but also pitfalls for the use of 3D printing for pediatrics. The capability to make designer procedures, we do that now, to actually design the procedure computationally, and then to print out the model for the procedure for the surgeons to use in the OR, as well as the surgical cutting guides. We can build patient-specific implants. That's sort of what's coming on board right now. And then I think you'll see patient-specific regeneration, printing cells and materials together. And if I had to make a prediction, I would say that leading medical centers, pediatric hospitals especially, if you want to be at the leading edge of treatment, in 5 years you're going to have your own 3D printing center for designing procedures, models, and instruments, as well as making patient-specific implants and materials. There are a number of pitfalls, technical pitfalls trying to expand the range of materials that we can use. Quality control. If you have one machine that can build thousands of different implants, how do you ensure quality and performance of these implants? That's a huge regulatory question, as well as the business model. But just to wrap up, I'd like to show you a video, which is actually the model I showed you at the beginning with the two splints and the use of 3D printing to treat pediatric condition. So if we can go ahead and run the video. I don't think we have the audio. Maybe the audio's not working. Of course, if the audio doesn't work, the video is not going to be much to watch, but basically I can maybe narrate it. This is Garrett Peterson. He had tetralogy of flow with also collapse of both bilateral bronchi, and he was the child for which we designed the resorbable implantable splints previously, and he was flown into the University of Michigan for the surgery, and Rick Oye, who's head of cardiac surgery at Michigan, was the chief surgeon on the case. And yeah, I don't know if we can speed up. Unfortunately, audio didn't work, but these are his parents who came in. And then if we'll just go through the surgery, you can see the actual implantation of the devices.
Scott, what was his prognosis before the surgery?
Well, he was so typically to treat tracheal malacia or airway collapse, it's done on a ventilator, and he had been on a ventilator for 16 months in a hospital, and he had never gone home. And actually at the point at which he was transferred, because many of these kids that get respiratory infections on the ventilator, their prognosis is very poor. So that's when the choice was made to fly him in and to have the correction of his pulmonary artery, as well as to implant the splints. So basically, the capability to do this, to implant the splints, you can see the implantation of the spiral splint here, that has basically saved his life, and this procedure has been done now 4 times with the resorbable splint and once with the permanent splint. So I'll end there, and we can shut off the video.
Could you comment briefly on your interaction with FDA with regards to this?
Yeah, so this was initially done under emergency clearance from the FDA. Basically, all of the patients that we treated were in severe condition. That was basically the only hope they had left. So the FDA cleared it, as well as the IRB at the University of Michigan. However, at this point, since we've done 4 patients under emergency clearance, the FDA sort of said, well, that's enough. You really need to move on and do a trial. We'll probably do a small feasibility trial, which is a new mechanism from the FDA to test devices. But this is actually a humanitarian use device, so it's less than 4,000 patients a year in the US. And I think that's another power of 3D printing patient-specific implants. It now becomes economically feasible to build these implants and these devices for humanitarian use applications, like in this pediatric case, and then we'll trying to gear up to run a phase 1 trial for the HDE approval.
Okay, thanks, Scott. Now we'll move to Raj, our first panelist.
Can we go back to the previous slide, please? I just wanted to thank Anthony and the organizers for having me here. It's been a revelation, and I seem to be one of the very few number of radiologists in the audience, and I'll make sure that there is a significant incremental representation next year. Can you go back to the previous slide, please? I just wanted to start with a little big picture of where 3D printing fits into our research program. I'm the Director of Imaging Research at Texas Children's Hospital, and you notice that we have a tissue-specific imaging biomarker development approach specific to pediatrics. And we break up the tissue functions into morphology, biomechanics, and function, flow, metabolism. And then image-guided therapy and processing and workflow are considered as facilitators, which enhance the individual boxes in each of the subspecialties. And cost-effectiveness analysis and healthcare provider education are common to all these boxes. 3D modeling and 3D printing fit into the last 2 boxes, image-guided therapy and processing and reporting. And they are considered as critical facilitators of each of these different boxes. So we don't consider 3D printing as a freestanding program on its own. We're constantly looking for incremental value provided by advanced modeling and printing in the whole process. I can tell you, I don't have a single cynical bone in my body, but we're very good at looking at the brutal reality of things. A lot of innovations have come out of this program, but in the last 12 years, there is only one which made it to product. And so that's a challenge that we constantly face, which is how do we enhance the impact beyond our own small local area? Because it's very difficult to get vendors interested in marketing a pediatric product. There's got to be either a professional, a personal, or some kind of philanthropic bent for them to get interested in pediatrics. So in the last couple of years, we've kind of changed our philosophy a little bit. We are now pushing all we can locally within the university, so we don't depend on the vendors to provide us critical tools to create these solutions. And then we make them shareable and then spread it around. So I'm going to show you one example of how we've used 3D printing for surgical simulation, and we created a process behind this which can easily be shared and replicated in other hospitals, and it does not entail any regulatory barriers. And we extend the same to guides, surgical processes. Anything short of implanting something into the body can be done in-house, and shared with other hospitals to replicate this process. So this is where there was a set of conjoined twins, which were unique in their extent of fusion. So the hearts shared the same pericardial cavity, the liver, the intestines, the renal system, all the gynecologic structures, they were all shared. And we went through a surgical planning process which involved creating inherent contrast within the different organs. So you notice in the heart picture, if you don't mind, I'll just point out a couple of things. You notice that one of the hearts has a lesser degree of contrast than the other heart, and that was deliberate in order to create distinction between the two hearts in the same cavity. Similarly, we put water into one intestinal tract and oral contrast into the other intestinal tract. So by doing that, we generated contrast between the different organ systems. So it took a total of 4 different CT scans to achieve this. And we also had to do individual angiograms, CT angiograms, to delineate the vascular supply to each of the individual pelvic organs so that we could determine how the organs are going to be assigned to one twin or the other. You notice there was a fair degree of complexity in the twins. 2 of the kidneys, 1 on each twin, were hydronephrotic and had an ectopic insertion into the urinary bladder. So we took all this data from different scans, integrated it into a single computerized model, and then we planned a 3D print which actually encompassed multiple organ information as well as vascular information into a single print. And you notice that this is the liver. We simulated the plane of separation of the liver, and then we incorporated vascular information into it so that the surgeon could see where the middle hepatic vein will be crossing over at the time of separation. Similarly, we created a plane of separation which included incision sites, location of the sternal margins, location of the liver into this model, and it was separable. And that was the ultimate result. There were no discrepancies between the surgical simulation and surgical findings. So this is a model which was built standing up to simulate how the twins would be. And so there were several decision-making steps that are involved, which I feel are easily replicable in a different hospital setting. I think this could be a standardized way to plan for complex conjoined twin separation procedures in the future.
Thanks, Raj. That's amazing use of 3D printing technology and a success story. So we'll move on to Tim, our last panelist.
Great. I'd like to echo my gratitude for being here and being part of this panel and the whole conference. It's an exciting time to be part of some of this new technology and how we can apply it to patient care and the unmet needs. As director of the HLHS program founded at Mayo Clinic, we have, in the last 5 years, launched the first FDA-monitored clinical trials for stem cell therapies in congenital heart disease. We currently have 2 ongoing clinical trials where we actually are delivering cell-based therapies into the hearts of infants at 3 months of age with hypoplastic left heart, and we now are doing intracoronary infusions in older children with failing Fontans. And it's been a really remarkable journey in the last 5 years to get this type of technology that has been pioneered in the adult world repurposed for the pediatric world. We believe that there's a dramatic advantage to putting these into a pediatric setting because of the regenerative response that you highlighted nicely. And I think that this is going to change dramatically the way we care for kids with congenital heart disease. If I could have my slide. There's one slide and one picture that I think defines our generation and our time. Anybody know what this picture is? Does anybody recognize this? This is a tweet that went out a few weeks ago with the caption, "There and back." 2 words, there and back, 3 words, I guess. This is SpaceX launch of their rocket into outer space with a time-lapse photo of it returning to the landing pad in a vertical position. Now, this was not a little bottle rocket that I used to play with as a kid. This is 15 stories tall that delivered a cargo payload into outer space that then delivered it, reversed itself, came back into the atmosphere, and landed about 12 minutes later in the exact same spot that it landed. This is creepy. And the fact that they did this with lower cost money than the NASA had spent in a shorter time period than anybody believed, and Elon Musk's vision that this was possible, to now be able to decrease the cost of space exploration, rather than that $60 million rocket on the bottom of the ocean, it's now refueled with $200,000 worth of fuel, ready to be launched again. The reason I showed this there and back again, because I think the definition of research and innovation that our program has embodied may be applicable to some of you. Innovation is the exact opposite of research. Innovation is converting knowledge into money. Research is converting money into knowledge. This was first stated by Jeff Nicholson at 3M, and I think is a profound statement. And if you think of the life cycle of research and innovation as being polar opposites of one another, you can now start thinking about how grant money can be a research grant, can lead to new knowledge, but then it can't stop there, that knowledge has to be converted back into innovation to drive new money. This has led to the creation of the Todd and Karen Wanick Family Program for Hypoplastic Left Heart, that its BHAG, if you will, isn't to go to Mars. Its BHAG is actually to delay or prevent transplant for all children with hypoplastic left heart. This is a research enterprise, but it's also an innovation enterprise that has an iterative life cycle, that's a cross-functional team of people that's iteratively learning from each other to try to figure out how to delay or prevent transplant for HLHS. And this really depends on regenerative medicine in our opinion, and all of the other technologies that get woven together to make this happen. So we have taken a pot of money, and we're very grateful to Todd and Karen Wanick Family Foundation for really investing in this vision from the beginning. And we've created research products, or product research, that has done over 250 whole genome sequences on families with HLHS. We've made iPS cells, or bioengineered stem cells, from all of those individuals. So we routinely have about 40 patients in the laboratory with beating heart tissue in the lab that's from our patients that we have whole genome sequencing on. We also have the capability of collecting cord blood on every patient diagnosed with HLHS so that we can process those cells and make it available for research going forward for the 2 clinical trials. And so this allows us to take that research money, drive new knowledge, and then convert that through clinical innovation, which really healthcare is a service organization that provides services to our patients. And with the right knowledge, we can accelerate that innovation, and we can incrementally, iteratively feed on itself so that we can have a new reality of applying this type of technology of regenerative medicine for a very specific need as congenital heart disease.
Great, thanks, Tim. That's a great way to conclude the session. I think all 4 of us will be around if there are any follow-up questions, but I'd like to thank all the panelists and Scott as my co-moderator. Thank you.
Thank you.